Dear friends, welcome to this edition of Basic Anatomy for the Allied Health Science course. We describe here in this chapter 4, lecture 2, uh, supportive cells of the nervous system, namely the glial cells. Now, we have in the previous video lecture discussed in detail about the structure of the neuron, the types of neuron, the functions of the neuron. But then the neuron by itself is not a standalone entity in terms of its complete performance. It has to have a um, armamentarium of support cells to optimize its functions. And that is where these non-neuron cells, namely the glial cells, come into the picture. Now let's put this neuron as a component of the whole histology of an area of the nervous system. You see, now there are so many cells. Now the neuron is identified. One cell size is much larger, and here in this photograph there are two neurons. But then you see the other cells we will need to identify. There are some cells which are colored blue as shown here. They seem to form some wrapping over the axons of the neuron. We'll see what it is in due course. Now there are another groups of cells which are attached some kind of a foot processes on the surface of capillaries. Then there are other minor cells also. We'll now move on to the identification of these glial cells. Now you see, here is an oligodendrocyte and it has a number of processes. Each process is going and wrapping around a part of the neuron axon of the neuron. This is an oligodendrocyte because one cell, glial cell, is wrapping around multiple neurons. But the same picture in the peripheral nervous system is replaced by the Schwann cell, with the difference that the Schwann cell will wrap only one single axon. That is a difference. But actual diagram here is the oligodendrocyte. The label bar Schwann cell is for the peripheral nervous system. Similarly, there is another cell which is showing foot processes which are attached to the capillary wall. Now those are the astrocytes. Next, we will expand this diagram and we will try to identify at least two more cell types. You see, there are tiny cells in close vicinity of the neurons. They seem to attach themselves to the neurons. These are the microglia. They are one is they are tiny and they have a lot of spiny processes. Other one is the ependymal cells. They are these cells, usually cuboidal or columnar. They are lining the ventricles and the central canal of the spinal cord. Wherever CSF is there, they are closely associated with it. Therefore, we move on to the discussion on a generalized function, and also we will go into more specific function of the individual types of the glial cells. By and large, we are already aware functionally they are support cells of the nervous system. Although they themselves do not produce electrical action potentials, they however help and play a role in optimizing the work of the neuron. So it can be all types of functions, as simple as protective function or support function, to nutritive support, nutritive regulation, then control of inflow of material into the central nervous system, some kind of a barrier, blood-brain barrier or a blood-CSF barrier, and also some kind of housekeeping where they detect neurons which are damaged or certain aspects of the neuron which needs to be certain outputs from the neuron that has to be cleared. So there seems to be a variety of functions to these glial cells. We'll go into the details one by one. Astrocytes. I've already introduced the astrocyte as a set of cells whose processes are attached to the capillaries. Well, to some extent here also there are some of the processes which are attached to the neuron, but the focus is on the capillary, what they do at the capillary. Their foot processes supervise the entry of material from the capillary into the neuron. So they keep a track, they keep a eye on the inflow material. So those are the food processes of the astrocytes. Therefore, this is a photograph where you can see beautifully the different astrocytes. You can also see marked in blue arrow one large cell, that's the neuron, and the one which are brownish are the astrocytes. The brownish colored cell and the arrow is black in color. You can also see here there is a blood vessel and the astrocyte is actually at, this is from an actual stained histological slide. Therefore, I have mentioned control and regulation of capillary permeability. Next, they form some kind of a periendothelial wrap, and to that extent they are principally they are very important components of this blood brain barrier by being closely associated with the endothelium. They also help in housekeeping function. They keep track of the ions and the nutrients in the extracellular space so that the electrical impulse transmission is optimized for the neuron. Next, there are two types of astrocytes, fibrous and protoplasmic. For your level of understanding, we will say if the cytoplasm is clearly visible and we will call it as a protoplasmic astrocytes. Very thin cytoplasm with hardly any details, we will treat it as fibrous astrocytes. So obviously there is some metabolic activity in the protoplasmic astrocytes, whereas fibrous astrocytes may probably be more concerned with the protection and things like that. Next, oligodendrocytes and Schwann cells, we will stick to one principal known function, namely formation and maintenance of the myelin sheath. Now you see, here is an axon of a neuron, and that's this actually the diagram is a Schwann cell. You can see how it is attached to the Schwann cell. In the next few animations, you will see how it wraps around. You see, it slowly goes around, and you can see the nucleus is moving at the wrapping edge. Now that's the formation of the myelin sheath. It turns around and around that, and finally forms a complete myelin wrap around over the axon. Although this is a animation showing the Schwann cell, more or less similar thing happens in the oligodendrocyte, except as already mentioned, oligodendrocyte will take care of about 20 to 30 axons at a time, whereas Schwann cell is individual. It goes to one single cell only, neuron only. This is another important function that this is to be noted, as I mentioned. Next, Schwann cell is present in the peripheral nervous system only, not in the central nervous system. Schwann cells are not only present in the myelinated nerves, it is also present in the unmyelinated nerves. But they don't produce myelin. It is believed to have some role in axonal growth and regeneration. That this is a very important function. Microglia are tiny cells in the immediate vicinity of the neuron. They keep a track on the health of the neuron. They will find out if any neuron component is damaged, and immediately they will convert themselves into macrophages. More or less, they are the macrophages of the nervous system, and they will make sure that these particles are removed or any unnecessary debris material is cleared away immediately. Macrophages of the nervous system. Ependymal cells are lining the ventricles as well as the central canal of the spinal cord. They are closely associated with production and circulation of CSF, and of course they form the blood CSF barrier. Satellite cells are present in the peripheral nervous system. I have given an example of the multipolar neurons or the autonomic ganglia. You can see the large huge cells are the multipolar neurons, and now those tiny cells in the immediate vicinity are the satellite cells. The function of satellite cells is more or less similar to that of the astrocytes, with the difference that the astrocytes are present in the central nervous system and these cells are present in the peripheral nervous system. Now that was an overview of the glial cells of the nervous system. Thank you.